Yeah, so thanks a lot for the invitation, which yeah, I'm very happy about. And yeah, I'm going to tell you something today about the chromosomal inversions that we have found in Malawi cichlids and what we understand and what we don't understand them. And I'm also very happy about any feedback that you can give. For us, it's kind of still, I think there's more that we don't understand than that we do understand. But yeah, and at any time you can interrupt me if there are questions, and if there's any problem with the connection or the audio, please also let me know. Yeah, I have quite a lot of introduction because I thought this is also a student seminar. So first of all, these are the people that are involved in this. So I started this work when I was still a postdoc with Richard Durbin, as you have heard, and now there are also several students and postdocs in my group involved in this at the University of Antwerp. And I think the very broadly the question we want to address is how do new species form? And in particular, in cases where this happens through natural selection. So of course, one enigmatic example of this are the Darwin finches, where you have these different ecotypes, where it's very clear that the speciation kind of involves an ecological component. So you have these beak sizes, which really make them adapted to feed on different prey items, either insects or different types of seeds. And what do we need to get such ecological speciation? Of course, we need some kind of heritable genetic variation. And the ultimate origin of variation is, of course, mutations. Mutations, for example, it causes a variation in the beak size or shape. Then we need selection on this variation happening. And we need some kind of reproductive isolation that that builds up between these ecotypes, so that kind of the variation kind of stays within these species and doesn't you don't get everything mixed up again. And so let's talk about the first ingredient, functional genetic variation. How do we get variation? So as I said, the ultimate cause are new mutations, but this is a notoriously slow process. So the per base pair mutation rate is only something between 10 to the minus 8 and 10 to the minus 9 per base pair per generation. And also this variation, when it's present in the population, it's constantly lost by genetic drift. And if it's functional or linked to something that's functional, then it's always also removed by selection all the time. So in general, it's thought that the genetic variation in population is relatively limited. So how do we get this variation that's necessary for this, especially for this very fast speciation as we see it in Darwin finches or in the cichlids we are working on? One potential solution for this is that you actually combine pre-evolved genetic differences between species. So if you have, species that are or have already diverged, and there was a long time for differences to build up between them. But if then you get hybridization between these genomes, as you can see here, can you see my mouse pointer?
Yes.
Okay, very good. Yeah, so here are species, they're kind of diverged. So this is shown by different colors. But if they still can hybridize, and then you get recombination, then you can actually get something where you have a combination of the different ancestries. And you can imagine that basically this picture that I showed at the beginning then looks a bit like this. So you have kind of different ancestral species, they hybridize, you get an ancestor, and from that you get diversification. I'm not saying that this happened for Darwin finches, but this is kind of a model that we are thinking about a lot. And it has been recently shown that this actually happens very often in nature, that you kind of get combinations of kind of old ancestry that gives you new adaptation, and not necessarily speciation. It can also be adaptation within species, but it's very common that kind of the genetic variation that is used is kind of, not new mutation, but it was kind of already present in some populations or species and then got combined together to give you a novel adaptation. And of course, what you need for this, the other thing is you need recombination. You kind of need that these different ancestries are actually combined by the process of recombination, as you also saw in this finch figure before. Okay, so the ingredients of successful ecological speciation, you need functional genetic variation. You could say, okay, we now know so hybridization, we could get this, for example. Selection, I think it's also easy to imagine that in a scenario like this, there is ecological selection for having beak that's good at insect eating or when it's good at seed eating, if these different resources are available. But the big question mark is how about reproductive isolation? And in a sense, there is a bit of a conundrum here. Of course, how important reproductive isolation is, this depends on the mode of speciation. And this is, in a case where there's kind of populations divergent allopatry, of course, it's obvious that they're reproductively isolated, but it's becoming more and more clear that, especially in this case of ecological speciation, this happens at least with some kind of contact. So in parapatry, where there's some kind of contact between the different populations or ecotypes. And then there kind of is a conflict of interest. So we just said before speciation, hybridization can be very useful to create new variation. And then recombination can be good to kind of combine these different ancestry into kind of new combinations. This is kind of on the left-hand side down here. But on the other hand, on the right-hand side, when these new kind of ecotypes are diverging, then recombination actually would break up these adaptive combinations that you have in different species, and it would kind of be detrimental to the process of divergence. And now the question is, how can kind of nature resolve this kind of conundrum that on the one hand recombination is good to kind of create new variations, but once you have them and there's selection for different combinations in different environments, then you kind of want to keep them together, even if there is still a gene flow. So here on the left is kind of good, on the right it's bad to have recombination. And one possible solution are chromosomal inversions. So a chromosomal inversion, this is if you have a chromosome and you have basically part of a sequence which flips end to end. And this can be actually quite large stretches of a chromosome where you get this, where it gets this. Usually it's kind of something in the when the chromosomes attach to each other and you have in meiosis that something goes wrong and this happens. So it's basically a type of mutation, but it's very different from a single nucleotide polymorphism. And one thing that's interesting about it, these inversions, even if they're large, they often have relatively small fitness effects because you still have all the genes. There's kind of no duplications or deletions, but it's just that their order changes. And the other interesting thing is that recombination is suppressed between the two opposite orientations of such an inversion. And this is just a short slide on kind of how recombination is suppressed. I'm, as Ovidio mentioned, not a biologist by training, so probably you don't know that better than me. But basically, there is a difference depending on whether the inversion includes the centromere or not. And things kind of work a bit different in the two cases, but the result is always the same, that actually the recombinant products of meiosis, they are usually not viable, and they don't contribute to the next generation. So basically recombination is strongly suppressed between the two haplotypes, whereas homozygous individuals, which have the same orientation, can usually recombine normally. And it's kind of increasingly shown that chromosomal inversions are very abundant in different natural systems. And there is lots of evidence that in different systems they actually contribute to local adaptation or speciation. There's this example of spring spawning and autumn spawning herring. So this is because a complex behavior, like the decision when they spawn. They usually are sympatric, but you have these two different forms. It has been shown that kind of the difference is mainly concentrated in a large inversion. You have cases of different life history strategies, like sneaker males in some birds. And you could also say that sex chromosomes are kind of something like this. You also have different kind of versions of a chromosome where recombination is suppressed. And then you can link together, for example, genes that are advantageous in the male sex on the Y chromosome. Okay, so how about cichlids? So we're studying cichlids, so it's kind of interesting. So what is interesting is that actually there's so far no evidence for the role of inversions in large vertebrate radiations. So in most of these radiations, there is kind of, although in other systems, it's kind of, they're very commonly involved in adaptation. In cichlids so far, there's not much evidence, partly maybe because the data wasn't there yet. So we wanted to look in these cichlids, and I think Malawi cichlids are an interesting system. It's you know, a bit like Darwin's finches, but it's on a much shorter time scale, and you have many more species. So more than 800 species, which is in less than 800,000 years. And this is also kind of shown in this graph. So this you won't be able to see the graph very well probably, but basically this should say that the maximum divergence between Malawi cichlids, which is kind of the divergence between species, is this histogram in orange, is almost 10 times lower than human-chimp divergence. So we, to our closest relative, are kind of 10 times more distant than the maximum distance between any of these 800 species. And you can also see that in gray, this is the diversity within species. And you can see the two distributions are really overlapping. So the divergence between species is not much more than the diversity within species. And we do know that all these Malawi cichlids can in principle hybridize with each other. So a question is there any mechanism that actually breaks up this the recombination of these alleles between from different species? So it's kind of reasonable to ask whether there are any such large inversions segregating between these species. So the question is, how can we find inversions? Okay, one kind of classical way is cytogenetics. So basically looking at chromosomes under microscopes, and this is, yeah, I mean, there are basically the first evolutionary biology was done like this. I mean, Dobzhansky, that's, yeah, much of his career was dedicated to doing this, and yeah, brought very, yeah, important insight, but it's also a very laborious process. And it only works for very large inversions. And usually you also need to know what to look for, because if the chromosomes are not deformed or anything, then you need some kind of fluorescent labels that you can actually see whether something swapped or not. Another possibility that, of course, becomes more and more viable is using long read sequencing, that you kind of have long reads that actually span these inversion breakpoints. And here is a little graph of what you would expect then. So imagine you have a reference genome, which is this kind of thing that goes from left blue to red on the right. This is just kind of showing the order on the chromosome. And then your sequence, when you have sequencing reads from a non-inverted individual, then you just expect that you map somewhere in the reference genome. However, imagine you have an individual which actually has a genome like this, where a part of it is inverted. So in the middle, this arrow is going the other direction. This is kind of the inverted segment. Then you would, of course, have kind of different reads. We kind of have different chunks from the genome. And if you now take these reads from that individual here and map them back to your reference genome, which does not have this inversion, then you expect that you get these split reads, like parts that map on the kind of left side and parts that map on the right side. So this is a way that you could detect inversions. But there also are a few issues with this. One of them is that you might need very long sequencing reads, because often these large inversions have very long repetitive regions. There's often, that's actually how these inversions often form in the first place, that there are kind of long regions of, for example, 100 kb of tandem repeats. And then everyone who has worked with long reads knows that getting reads that are longer than 100 kb is not that obvious. So it's still challenging to do that, at least in some cases. And also it's quite expensive, of course, to get long read sequencing. But how about short reads? So for Malawi cichlids, for example, we have lots and lots of short reads for 2,500 samples from 270 species. So the question is, can we actually ask whether there are inversions from the short reads? So with mapping, that generally doesn't work, because of course, if you just have short reads, they will map in the reference genome wherever they are in the reference, even if there was an inversion. So you don't see it directly, but there are actually some indirect signals which come from basically the effect of inversions just on recombination, and the fact that recombination is suppressed in this region. On the one hand, there could be some phylogenetic discordance. So yeah, anyone who works in, yeah, if you know phylogeny a bit, you know there is usually this idea that gene trees are not necessarily the same as the species tree. So you can have local variation in relatedness pattern because of incomplete lineage sorting, because of, yeah, because of the fact that some individuals don't find the common ancestor within the species, so they can coalesce first with samples from other species. And this is something that, that's very common, and especially Malawi cichlids, it's very common because they're so closely related. But you generally expect that this happens on a relatively short genomic scale. So that, like, this happens, for one gene, but if you kind of take a larger window and a larger window, then you should converge to the genome-wide tree. But of course, if in an inversion, and that's because of recombination, but if there is an inversion and recombination is suppressed, then this is not necessarily what you expect. You could expect there's a large region where you don't actually see this, where that is this convergence to the genome-wide tree. So basically, you could find large regions where the local relationships are different from the species tree. And then also the patterns of genetic variation could be different in inversions just because of this reduction in recombination. On the one hand, if the inversions are young, then this did likely increase in frequency quickly because of positive selection. Then you expect reduced diversity. And if they were transferred across species, then you can actually expect reduced divergence between the species or the above mentioned phylogenetic discordance. If inversions are sex determining, then of course, there also is an expectation that you get increased diversity in the heterochromatic sex because they have the heterozygous for the two different haplotypes. Okay, so in Malawi cichlids, this is kind of the general phylogeny that we know of. So there are these benthic species, which are kind of an outgroup to the rest. Sorry, not benthic, these pelagic species. Rhamphochromys are kind of open water predators, and Diplotaxodon open water mainly plankton feeders. And then here you have rock-dwelling cichlids, a species that goes into rivers, and this is a relatively diverse group of benthic species. So each of the branches you see here are actually major groups, and each of them has many species. Here you can see the number of, sorry, these two need to be swapped. So this is the number of genera in each of these groups, and this is the number of species. So each of these groups has relatively many species, as you can see. Generally, these groups are relatively clearly defined, but we know there is lots of incomplete lineage sorting, so that local gene trees, they can look very different from the species tree. Now, one thing we did, and I have to confess that at this point, the idea wasn't even to look for inversions, but we kind of stumbled upon this. The idea was to look for local phylogenies, going across the genome and ask how are species, related to each other in windows. And we use this program Twist, and I don't think I can go into detail about what exactly it does, but in principle, it looks for, it kind of gives you a score of how common a given topology is in a window between a group of 4 species, for example. In this case, we ran this, the 4th species was an outgroup, so we were kind of looking at 3 other species, and we asked how they are related. And after apology, the blue species, something got wrong in the conversion of this, so it's actually does have a head and a fin here. But this is the genome-wide pattern. This is the rock-dwelling species are assisted to the benthic, and the pelagic are an outgroup. And then there are 2 possible alternative topologies. And this one is called introgression because it actually, this corresponds to a known gene flow between the benthics and the pelagics. And this here is one of the chromosomes, and you can look across the chromosomes and ask how often do you see each of these topologies. And you will see that this blue one, the genome-wide, this is basically the one that if you build a genome-wide phylogeny, this is the one that we get, like a consensus, you could say. We can see that this is actually the most common one, the blue one. And the other ones, we expect them both to occur because of incomplete lineage sorting. And in addition, we expect to occur the introgression one because of this hybridization that we know of. And we see that the green one is a bit more common than the orange. But in general, this is kind of more or less what you would expect. And now I'm going to show you exactly the same thing, but for all chromosomes in the genome. And this is how this looks like. Again, it's a bit more trees now because we use one more taxa, so we use 4 taxa now. And these are the different chromosomes here. And again, you see that the species tree is green now. It's generally the most common one in each of these windows. The windows are larger, so there's less noise. But now you see this extremely striking pattern that on some chromosomes, you consistently see a different topology between these ecomorphological groups. So you see that consistently here on chromosome 9, you get these topologies where kind of the benthics are a sister to this pelagic group Diplotaxodon, And then on some other chromosomes, you get consistently other patterns like this. And this is just something that in a normal population genetic model, if recombination is happening, you cannot really explain it unless this haplotype is fixed within like a very short amount of generations. But we know that this is not the case, because it's actually, because the diversity patterns are not consistent with this. So our only explanation for this really was that there is something that suppresses recombination in these regions, and chromosomal inversions is kind of the most obvious explanation. So can we find any evidence for this? So the first thing we tried for this is doing, actual cytogenetics that I was mentioning before. So designing probes. So this is on one of these chromosomes. We design probes on either side of the inversions, and there's one probe inside the inversion. And then we can look at two different species that we expect to be different for the inversion, and do this cytogenetics. So it was actually the cytogenetics group at Sanger that did this. And you can, if you carefully look at these chromosomes, you can see that there actually is a pattern which is consistent. So you can see that in this one species, this looks like the reference. So you have the red and the yellow very close to each other, and this magenta one, which is far. But in the other species, you see that it's actually, so there was a different dye used for technical reason in this one, but it's again this locus. But you see that this locus and the red one, they're close to each other, and the yellow is very far. So in this case, it was consistent with the with really that there is an inversion. We also tried this for the other inversions, but actually we got many off-target signals. There were lots of technical problems with the cytogenetics. So only for one of them we really got strong evidence. We also tried to use long read assemblies for some species, and could also find evidence for one of the inversions. And actually, so here this is a read mapping against the reference, and you can see that you get these kind of split mappings. And actually here this is consistent with like a double inversion, that you get kind of two segments which inverted like that. So that's kind of good. So this is really strong evidence that this indeed is an inversion. But I also have to say that for the other inversions, we actually either got no evidence or negative evidence. And the reason is, on the one hand, technical challenges with these assemblies. They often actually break where this inversion breakpoints are supposed to be. But it's also that at the time actually we selected the wrong species. If now, knowing what I know now, most of these species, we don't expect them actually to be different from the reference. It's only these first two that we expect to be different, and the others we expect to be the same. And yeah, that's about long reads. And now I want to talk a bit about trying to interpret kind of the evolutionary history of these inversions and what they are doing. And I have this plot here, and this is probably on the left-hand side here. And many people find that I really like this plot, but many people think it's very complicated. So I will spend some time in explaining it, because I think it's a good way to looking at these inversions, or kind of generally to looking at the evolutionary history. So what you see here is actually simply a matrix of the pairwise genetic differences between different groups of Malawi cichlids. And the color scale here is that the brighter this is, the more different they are, and the darker it is, the less different they are. So things that are relatively closely related are relatively dark red. And basically on the X and the Y axis are the different samples. So on the diagonal would be kind of a different samples from itself. And the off-diagonal entries are differences between different samples. And you can see that the samples are actually ordered by the genome-wide phylogeny. So you kind of see that the ones which are close to each other are generally more closely related. And what we see here is, on top, you see the two different pelagic clades, the ones that are most different from the rest. You can see that they are relatively well defined. Then here we have this big clade of rock-dwelling Mbuna, also clearly defined. And then we have this large group of benthics here. So this is this is large because it's where we have the most samples, but there are also many different species. This is just you know, a subset of the actual species that exist in this group. Okay, and kind of what is important is that in this group there are actually subgroups which can be distinguished. It's not so clear here from these plots, but maybe if you look carefully, you can see that these are a bit closer to each other, and then the ones in the middle here are a bit more different. These are, if you do phylogenetic reconstruction, you find these 3 different groups: the deep water benthic species that generally live in the deep water, the shallow water benthic species, and the Eutaka, which are semi-open water plankton feeders within this group. So you can distinguish these 3 subclades. And what you can also immediately see here is that these off-diagonal entries, this is the difference, for example, in the clade Diplotaxodon, this pelagic species, and the benthics here. And you can see that the benthics are actually a bit closer to Diplotaxodon than they are to Aramphochromis, the sister group of Diplotaxodon. And this is what I was mentioning before, where we kind of have evidence that there was gene flow between these 2 groups very early in the evolution. So probably it happened in the ancestral branch of all these benthics and all Diplotaxodon. Okay, so now that I have, now that you know how to interpret this plot, we can, of course, so this is the kind of the genome-wide patterns, or to be precise, actually the genome-wide patterns in these regions which don't have any evidence for having something like an inversion. And of course, now I can make exactly the same plot, but in the inversion region. We can ask, you know, does it look the same or does it look different? So, you know, how is the evolution different? And this is what you see here. So on the top here, you see exactly the same as before. So this is the genome-wide non-inverted pairwise differences between different samples. And at the bottom, it's kind of the same order of samples, but you see the pairwise differences in the chromosome 11 putative inversion region. And you can immediately see, yeah, that things are very different in this region, right? And I will actually come back to this and interpret the differences. But for now, I just want to make the point that the bottom here looks very different from the top. I think everyone will agree with this. It's qualitatively a very different pattern. And we thought that actually, so we thought a bit of, yeah, we put a bit of thought into how we can actually kind of more exhaustively detect such kind of putative inversions, because we did this twist and we kind of found some things, but for twist you need to make a choice of, you know, 3 or 4 or maximum 5 species. So it's a bit arbitrary, and it's not clear how to define, you know, what you expect depending on, it always depends on, you know, the exact evolutionary history of these inversions, what you expect. So we said, rather than making such kind of a detailed kind of assumptions, we just want to actually look across the genome for regions where we consistently find, you know, such weird things, where we find that actually the, you know, the local relationships are very different from the genome, no matter in which exact way they are different. So the idea was to kind of develop a robust method which can actually look for such patterns down there. Just regions which are like weird, different from, you know, the genome-wide average, consistently different. And what we did for this is we took distance matrices in 100 kb windows along the genome, and then basically wanted to cluster them to see differences. But of course, it is also clear that there is kind of lots of collinearity in these matrices, because of course we know that the samples are in principle related by a, you know, by a graph, so a tree plus some gene flow edges probably. But of course, this means that lots of the information is correlated in these matrices. So to remove these correlations, we actually ran a PCA, a principal component analysis, on these matrices, and then we look at the first principal components of this. And this is what you see here. So this is a PCA on the left, and you might be used to look at PCA where kind of each dot is a sample. But this is not what you see here. What you see here is each dot is actually one of these windows in the genome, and windows with pairwise differences of all samples. So you can do something like this, and then the idea is to run some kind of machine learning classification where we can actually classify, you know, different windows into different categories depending on these PCAs. And when we first tried this, it actually was not very informative. It really it mainly kind of differentiated outlier windows, which probably were, you know, regions of low quality in the genome or repetitive regions. But after we put in a stringent filtering for kind of removing regions where, you know, where we have filtered lots of the genome, or regions where, or kind of outlier windows, when we then run such a clustering algorithm, we found that this actually works very well in identifying these inversion regions. So it identified the one that we had already known and a few others. And what I show here is for the one that we had already seen in Twist. This is always the plots is kind of the going across the chromosome. On the x-axis is the chromosome. And the red lines are kind of the known inversion breakpoints that we kind of know either from Twist or from long read. So it's not from this analysis. It is totally independent of this clustering. And then the red and the gray, and I'm sorry, this is, I think, not so easy to see maybe, depending on your screen, but the red and the gray, that's the clustering. So that says for each window across the genome, whether it belongs to one cluster or the other. And we see that actually this works very well in identifying these inversions. So when we run this clustering on chromosomes that have inversions, we really see this very clear cluster that are, you know, have this spatial correlation. We get a long chunk of consistently one that's in one cluster. And if you run this on chromosomes that don't have an inversion, we just, you know, the dots are kind of random, but it's not, you don't get this kind of sorting of the colors between in the inversion or outside. So we find that this works well, and yeah, in some cases there's still some outliers. Plotting the PC1 here is actually a bit arbitrary. You see that actually on PC1 there is not much difference between inside or outside. In this example, we see it's actually PC2 that loads on the difference between inverted and non-inverted windows in the genome. But in other cases, it can be higher PCAs, or it's actually a combination of different PCAs. So that's not always clear, but what we see is that this method works very well in kind of finding these inversions, like going in totally naive, not knowing whether there is anything or not, just looking for consistently weird regions in the genome, I could say. Of course, after this, you want to kind of look further what actually happens in these regions. But before that, I also want to mention that we also try a different method which works with windowed PCAs, but this is basically very different. It's also a PCA, but it's before we saw a PCA on pairwise difference windows, and this is a PCA on SNPs in windows across the genome. And here, what is plotted here on the x-axis, again, the chromosome. On the y-axis, you see the first principal component, and you see that actually the first principal component looks very different in this inversion region than it looks outside. Outside, it kind of wiggles around a bit, and there's not so much variation. But then we kind of see this very clear three groups, and these are basically the three inversion genotypes: homozygous reference, heterozygous, and homozygous like inverted. And the other thing that's very clear is that actually the amount of variance explained by PC1 is kind of hugely increased in this region. So really the lots of the divergence in our sample is explained by the difference between these inversion haplotypes.
Sorry, Hannes, can you explain me, how do I read these different, whatever, Turkish or whatever, this little color, because it's in the top and then the middle, it's in the lowest category. How do I read this? Do you have multiple breakpoints or? Okay.
The colors here, you mean, or?
Yeah, you have this in the middle. This is what I thought. This in the middle is the third.
Yeah, that's probably the heterozygote, and indeed the fact that this goes down here, that's probably like a double recombinant.
And in the upper thing, you also have one of these colors. So
okay, sorry. So the colors is actually which genus these are from. So what we see is that actually some, the inversions are kind of segregating across species. It's not fixed in one species, but individuals of the genus which is red here, Olonocara, they are predominantly have this genotype, but then also some red individuals which have the other inversion genotype.
Okay, I get it. Thanks.
And there is one, one species from this green genus which has this genotype. Yeah, sorry for not explaining the colors. And then below the colors are totally different. This is PC1, PC2, PC3, the amount of variance that's explained by them.
Before you move on, I have a question also. Each line is a different individual from a different genre. Like
each line?
Each line, I get the colors that there are different species, but each line, what exactly does represent? Each line is a different individual. Okay. Yes. And there are like more than one individual per genus. I don't know if... Yeah, there's more than one individual per genus, and in some cases there is more than one per species also. Okay. Yeah. Thank you very much. Yeah, welcome. One more question, Hannes. Yeah. Right in the middle, the green one goes down and up again. Yeah. Yeah, so that's we think that this is a double recombinant. So this is an individual which doesn't have the whole inversion haplotype, but apparently it recombined. Yes. So one of the two haplotypes has a recombination. Okay, thanks. But what is actually quite interesting is that we would have expected to find more of these double recombinants, because this is relatively long, this inversion, so there could be double crossover. But actually, I think this is the only individual where we found it. Partly this might be because, as we will see in one of the next slides, often they're actually fixed within species. So the fact that they're kind of variable within genera doesn't mean that they're variable within species, because sometimes the genera are actually not very well defined. They're actually not even monophyletic in some cases. But still, it's kind of surprising that there is so little recombination apparently between the haplotypes, because in some cases they are polymorphic even within species. Yes. Okay, and if we now, here the PC is kind of in SNP windows, again in 100 KB windows, but we could, now that we kind of know approximately where the breakpoints are, of course we can also take the whole region and do a PCA on the whole region. And this is again a PCA of samples, PC1 and PC2. And then we see that actually on PC1, for this inversion, it's very clear that what we get here are the haplotypes. So you need to believe me now that this is what we see, but we kind of know that, yeah, we see that samples of the same species, or they kind of cluster here by their genotype in this inversion. And we can use this to genotype basically all our samples that we have for each of these inversions. And when with this, I kind of want to get back to this plot again that I showed you before. So on top, this is the pairwise genetic difference between samples in the non-inverted part of the genome. So this is the genome-wide average. And down here, this is in this chromosome 11 inversion. And yet now what we see here is actually that these clades up here, Rhamphochromys, Diplodoxodon, and the rock-dwelling Embuna, they are relatively similar to the rest of the genome. But down here in the benthics, it's totally different. So we have, whereas in the rest of the genome, they're very similar to each other, we have these two relatively divergent haplotypes here in these benthics. You see that one of them has actually very low diversity. And the other interesting thing is, that we have these lines here. If you look carefully, there are some samples which actually don't fit this pattern. So it's kind of they're not totally monophyletic, these different groups. Sorry, this is the same plot as before. And yeah, so what's going on here? Why, did this introgress from other species? And another thing that we see is, that actually some of them are kind of some halfway the distance of the two groups. This could either be that they are recombinants and they only have half of the inversion haplotype. But actually, if we look at heterozygosity, then we find there are some samples which really have very strong excess heterozygosity. And this is consistent that they're actually heterozygote for these inversions. And specifically, if we look at one of the species where we have many samples and we have kind of a good designation of whether they are male or female, we see that there actually is a 100% correlation of the heterozygosity in these regions on the y-axis heterozygosity. And here this is in females and males. So we see that males have a much higher heterozygosity than expected. And basically what this means is the males are heterozygote for the inversions and the females are homozygous. So it looks like in this species, this actually is a sex determination system. This is an XY system on chromosome 11.
And we kind of have evidence for this in kind of 3 species, which are not sister species here. And, but in this other group here, for example, this haplotype is fixed. And it's kind of, I try to wrap my head around how to interpret this. But the other kind of very strange pattern, and I think, I'm afraid that this, yeah, it's what I find very interesting is that also if you look at the relationship of these 2 haplotypes to outgroups, you also see a very different pattern from the rest of the genome. So here we said, okay, there's gene flow with Diplodoxodon, so they're a bit closer to Diplodoxodon than to the other outgroups, and this pattern is even stronger for this X haplotype. Remember, I call this X haplotype here, but in this species, it's actually not a sex determination system. In this species, it's fixed. And I call this Y haplotype, but it's actually fixed here. It's not sex determination. It's only XY in these species in the middle here. Yeah, so this kind of shows this pattern even stronger, that they're closer to Diplodoxodon, similar to here, but stronger. But this other haplotype, they're totally equally distant to all other groups in this. And yeah, I don't think I can go into detail, but my explanation is that this is an old sex determination system. Actually, the common ancestor of all these species had this XY system, and there was less integration on the Y haplotype because they didn't have this system. And so the Y haplotype is just kind of void of integration, and that's why it's equidistant. And you had more integration on the X than on the Y, which you can also expect given the smaller effective population size. And yeah, this is the hypothesis. I think I need to speed up a bit, but just to tell you, okay, the same thing that I just did now for this chromosome 11, you can also do it for the other chromosomes. And basically what we see is that 3 of them have a pattern which is qualitatively quite similar to each other. It's generally the deep benthic versus the shallow benthic species, with a few exceptions in between. But in general, it's these groups which are kind of highly divergent. So it seems that this kind of evolved in the common ancestor and on the branch that leads to the present-day deep and shallow benthic species. So it's yeah, it's possible that actually these inversions played a role in suppressing gene flow in this kind of initial speciation between the common ancestor of all the deep benthic species and the common ancestor of all the shallow benthic species. Chromosome 9, on the other hand, looks very different. Actually, it has this very strange pattern where there are kind of samples which have it and which don't have this one haplotype. And it also turns out that it only occurs in heterozygous state. There are no individuals where it's kind of fixed. So we think that this is another sex determination system that's active in other species. So the chromosome 11 is in some species in the middle here, and down here it seems that many of these species have a XY system with this chromosome 9 inversion. But there also is another interesting observation that I will mention later about this chromosome 9. Okay, I think I will skip over heterozygosity because we are running out of time. But yeah, heterozygosity is as expected in hets, it's increased, and then the derived haplotype is generally decreased. If you look along these inversions, this is diversity statistics along the inversions. And yeah, this is more interesting for people that know a bit about inversion theory. So generally you expect something that like a U-shaped pattern of divergence between the different haplotypes and the upside down U-shaped pattern of diversity in the inversions. And the reason is that you expect that there's almost no recombination at the breakpoints of the inversions, and that there is kind of more, that a kind of double recombination or something is more likely to happen in the middle of the inversion. So you get more gene flow between the inversion haplotypes in the middle, so there is less divergence between them and there's more diversity within them. But this is just to say that although with this diversity statistics, it's very clear where the inversion starts and ends. So you can see this very clearly. The pattern is different inside from outside, but there is no clear U-shaped pattern here. So along the inversion, it's kind of very, very similar, which suggests that there is kind of not much recombination going on between the two haplotypes. Yeah, the question is why? Either because they're relatively young, so this process didn't really play out. So I think in Drosophila, this is what you usually see. right?
Yeah, that's correct.
Yeah, so we don't see this at all here, either because it's younger or because it's actually, in most cases, not polymorphic.
In Drosophila, you can distinguish is the old inversions have this pattern that you describe, and the young ones look like yours. So
yeah. So maybe it's just the age. Yeah, these are of course younger than the radiation, so they are probably on the order of less than 100,000 generations, 30,000 generations. Okay. So the last thing that I wanted to show is we also, of course, we're interested in whether there's selection happening on genes in these inversions, whether they're doing something. And one thing we were asking is whether there's kind of a differential fixation of non-synonymous variants compared to synonymous variants, which is kind of what you kind of don't expect in a neutral model, which you only expect if there is selection happening on these non-synonymous variants. And we kind of developed a test for this, which basically is very similar to some kind of DNDS or PNPS rather, but it kind of controls for the background. So basically it's just the non-synonymous over synonymous ratio for SNPs which are inversion diagnostic, so which means SNPs which really kind of are differentiated between the two inversion haplotypes, compared to random SNPs which are not kind of differentiated between the inversion haplotypes, which are just any kind of SNP, any kind of polymorphism. And when you do this is in this plot here on the left hand side, you see the SNPs which really are very inversion diagnostic. So they are really basically the SNPs which are fixed differences or almost fixed difference between the two haplotypes. And on the right hand side, you see the ones which are kind of less diagnostic. And you really can see that there's a strong excess of this statistic for the ones which are very inversion diagnostic. And on the next slide, I'm going to show this for all chromosomes. So see that especially for some of the chromosomes, there's a very clear pattern that this is increased here. Some people might wonder why this is decreased on the other side. I have an explanation for this, but maybe I move on now. So because are we supposed to finish at 6, including questions? So I need to stop soon, right?
Don't worry so much.
Yeah, we are flexible.
Okay. Okay. But the point here is it seems that, these kind of the evolution of these inversions, this violates clearly a neutral model. So there seems to be selection going on. But when we actually look for enrichment, whether there are specific genes, we didn't find any strong candidates. So we are not clear. We actually don't have a good hypothesis yet of kind of what's driving this selection. But this is very, very recent results. So this is something that the postdoc is currently looking at. But of course, kind of the interesting question with inversions, of course, is always, is there any phenotype? So if we're in the selection, there must be some phenotype. I mean, one phenotype we saw that's very clear. Sex is, of course, a clear phenotype. But other than that, there is some evidence for example, they did a GWAS study on a behavioral phenotype, which is the males build some mating platforms, and this can be either a pitch shape or a bower shape. And when they did a GWAS on this, then they actually found this kind of big peak on chromosome 11, and this exactly falls into our chromosome 11 inversion. But I think it's also difficult to interpret this really as evidence that inversion does anything causal, because there is a strong phylogenetic bias, because these species that build these castles and bowers, they are kind of still have some phylogenetic correlation, although there is some, there is some parallelism that it kind of evolved several times, but still, it's often not clear. Well, it's a bit complicated because I was telling you that, for example, on chromosome 2, we have this, it's generally deep versus shallow bentics that are different. And this is shown here on the right-hand side. So you see one inversion haplotype is in red. So this is a species which are fixed for this haplotype in dark red. And then the other one is in black. And then the ones which are light red are the ones which are polymorphic. And you can see that in general, chromosome 2, you have this deep versus shallow pattern. But then here and there are some shallow species which actually also have the deep haplotype. And the one question you could ask is, are they kind of ecologically more similar? And actually, this is often the case, that they're ecologically more similar, but it's hard to say that this means that the inversion haplotype gives any ecological adaptation, because I think it's more likely the other way, that if they are ecologically similar, they're more likely to be in the same habitat, and then they're more likely to hybridize and actually transfer the haplotype from one group to the other. So I think it's more likely that's what's happening. But that said, we are kind of still looking for any phenotypic differences that we could find that are associated with this haplotype other than sex determination. And one potentially interesting thing is, there might be balancing selection, because sometimes we see the species are clearly far from Hardy-Weinberg equilibrium in some species. And we actually have a PCR assay where we have transposons which are kind of in the breakpoints of the inversion, but only on one of the two haplotypes, and then we can use this to actually type individuals quite cheaply and easily. And we did this on an aquarium population, and there we actually find a very strange observation that of this aquarium population, 20 of 22 individuals were heterozygous for this chromosome 13 inversion. So most of them are heterozygous. So it's very far from Hardy-Weinberg equilibrium. How can this be? Of course, if they mate, you expect that you get a restoration of Hardy-Weinberg frequencies. And the only one of the explanations that we could come up with is that actually these haplotypes carry recessive lethal alleles, that actually somehow there is strong selection for these to be heterozygous, because there are some recessive alleles which are very deleterious on the haplotypes, and if they are exposed, then you get some non-viability. And currently we're setting up crosses between them to see whether this is happening and whether there are eggs which are dying or don't develop. But I also have to say that this is something that happens in the lab. In the wild, actually, you don't get such high heterozygous fractions. So there this might actually be some kind of deleterious allele that got to high frequency when these species got into the lab and were bottlenecked there. So this is basically it. The conclusion is it all looks very complicated. It's kind of exciting, but we are far from understanding most of it. Generally, many of these haplotypes many of these inversions separate the deep versus the shallow benthic species. So maybe they played a role in the divergence of these groups, kind of to impede gene flow between these groups when they originally formed. What is weird that sometimes the relationships to other groups change significantly. So we are, in some cases, I think an explanation is that at least one of these inversions is an old sex determination system in the whole group. Possibly it could also be for other reasons, that they're kind of impede gene flow from the other groups when you have these inversion haplotypes. That's basically what I had to say. I think this inversion is there's potentially something very interesting going on, but it's not as obvious as it's in some other systems, what exactly they are doing here. And what we're doing now is we want to try to model their evolutionary history. We want to kind of type them in large amount of samples to really understand how their frequencies were changing within species, and also how they were transferred across species. How did they actually, get to the distribution that they have currently? With this, I want to thank my collaborators, in particular Richard Durbin, my former supervisor, and you for your attention, and I'm happy to take questions.